is key, right? So what do we take from here? We can see that there's some consistent evidence in the epidemiologic context for an association between higher UPF consumption and abdominal obesity, type 2 diabetes, chronic kidney disease, and overweight and obesity. But nothing really for cancer. You know, there's this non-significant increase in colorectal cancer based on just 4 studies. And then there's this all cancer mortality, null association. But really, there's a lack of evidence in the cancer space as not directly highlighted, but highlighted in the absence of it being presented here in this Lancet paper. So when I first decided to jump into studying ultra-processed food in relation to cancer risk, we decided to use the NOVA definition. And we decided that because it was the most widely adopted definition, 5-6 years ago when we started thinking about how to operationalize this in our cohorts. And I remember reading this paper in Public Health Nutrition by Dr. Monteiro. And I'm not going to read the whole quote. You all know, the NOVA definition from the Brazilian group, but it essentially says these are formulations made mostly or entirely from substances derived from foods and additives with little, if any, intact group 1 foods. Now, group 1 foods are shown in green up there, and I apologize that this is so small, but I know that you are all very familiar with the NOVA classification system. So these minimally processed or unprocessed foods, they include things like eggs and milk, but also red meat, right? I'm a cancer epidemiologist. I study colorectal cancer. Red meat is an established risk factor for colorectal cancer. You go down to the other groups, you have the ultra processed group. These are your industrial formulations, things like packaged breads and cereals. Again, I'm a cancer epidemiologist. A major source of whole grains and something that we consider to be an established protective factor for colorectal cancer is whole grains. So, trying to think about what does this mean? And this again gets back to one of those core principles, what's harmful, what's helpful, what's processing, what's formulation. These are all important things to think about. And applying this definition consistently is challenging. So I'll get a little bit more into that. But before I do that, I just want to give you, before I show you my data, I want to give you the epidemiologic landscape by highlighting 2 very large prospective studies. First, data from the UK Biobank and then the EPIC cohort. And I'm doing this instead of showing you meta-analyses because at least here I can say that within the study, it's internally valid, right? They use the same definition. We're not trying to meta-analyze across a bunch of different people's definitions. So in UK Biobank, they had sufficient dietary data to do this for about 200,000 participants. They were middle-aged and older. They completed this Oxford WebQ, which is essentially a 24-hour dietary questionnaire. And they were followed from 2009 through 2021. So you have about 10 or so years of follow-up for cancer outcomes. And their main analyses that they present in the paper are for percentage gram weight. Now, this plot here, it's a little small, but what I've highlighted in red is the top contribution to group 1 grams, and then lower down the top contribution to group 4 grams. And why this is important is because the denominator of percent grams is going to be driven by group 1, right? So highlighted there is tea and coffee, right? So the largest contributor to the denominator is grams of tea and coffee. And then the largest contributors to the numerator are carbonated drinks and other fruit-based drinks. So this is essentially a beverage ratio, right? And you see these smaller contributions for all of these food items. And you can look up the paper, but I think that it's important to know what is the percentage gram variable that we're looking at? What is it representing? When they looked at it in relation to cancer outcomes, they looked at all cancers. And you can't see the estimate up there, but they found that percentage grams of ultra-processed food, the highest quartile compared to the lowest quartile, was associated with a 7% higher risk of all cancers. Now, nobody needs to be told that all cancers is not a very useful outcome, right? Cancer is a heterogeneous disease. The etiologies for different cancers vary wildly. And so I, don't put much stock in this. But what they also found, the only other positive or the only other significant associations that they found after adjusting for multiple comparisons because they ran so many tests was this sort of odd, potentially spurious inverse association with head and neck cancer, and then this positive association for ovarian cancer. Now, ovarian cancer is not known to be a diet-related cancer, and so this piqued my interest, and I'll show you a little bit more of our own data on that shortly. But for the main ones, colorectal cancer, the main breast cancer, what we think of maybe being diet related, they didn't observe associations in UK Biobank. Switching over to EPIC. In EPIC, this is much larger now. We have nearly 500,000 participants, similar age range, similar length of follow-up, and also using percentage gram weight. I don't have illustration to show you sort of what's contributing to the numerator and the denominator, but again, it's very heavily weighted toward beverages, both in the numerator and the denominator. And here they're showing all the NOVA subgroups. Again, it's small, but the main takeaway here is that there were not many associations observed for NOVA group 4. The only one that they saw was a positive association potentially for head and neck cancer in their substitution analyses, but this was largely driven by substituting NOVA group 1 foods for NOVA group 4 foods, as opposed to being driven by intake of NOVA group 4 foods specifically. But again, the most you can really take away from here for those the hazard ratios, which are the colored horizontal lines in that last column for NOVA group 4, it's showing all of the different quartiles. Some of them are to the right, which means potentially positive association. Some are to the left, which means potentially inverse associations, but they're kind of all over the map. And at best, we can say there might be some modest associations here, but we're not going to be seeing with larger numbers, any 3, 4, 5-fold increased risk of cancer with UPF. Okay, so what are some of the challenges that my group has been thinking about? And again, a lot of these challenges that we've been discussing over the years are well reflected in the core principles of IFINs that were presented. One thing we think about, because our cohorts largely rely on dietary questionnaires, is measurement error. We have systematic measurement error, which includes things not being queried about, right? You can only ask about 120 or so items, so some items are going to be missed, and that may or may not be important for your given population. And then we also have random error, and random error applies particularly to things like recalls, where when people are reporting on one day, their dietary behaviors might change from day to day. And so you really need serial assessments to be able to get that more detailed capture of diet. Now, here I have a snapshot from the cohort that I mainly work in, which is the NIH AARP Diet and Health Study. And I've highlighted the questions that we have on cereal, because cereal is going to sort of make a guest appearance in some later data that I show. But it's one thing that was asked with a fair amount of detail in our FFQ, and in particular, trying to get at fiber intake, right? So it was we asked about different high fiber cereals. And when thinking about NOVA classification, we have the high fiber cereals in that pink row there, which includes Cheerios, shredded wheat, Raisin Bran, Bran Flakes, granola. Now, these are likely assumed to be UPF, but if it's homemade granola, it wouldn't be. I've seen somewhere that shredded wheat isn't supposed to be categorized as NOVA 4, but Cheerios are. And so it's just sort of, how do we consistently apply this to our data? FFQs for the most part, lack detail on extent and purpose of processing, which again is key to that original NOVA definition. And then even if we did have the detail, we lack the databases to do any sort of estimation of ingredients or food additives or contact contaminants or things that we might be interested in terms of studying carcinogenesis. Some other challenges with the NOVA definition is it's a broad umbrella, even for something like cereal. So thinking about that sort of harmful core principle, you know, on the left, we have Fruit Loops with 15 grams of added sugar, unsweetened plain whole grain Cheerios with 1 gram of added sugar. They're both UPF. They're considered the same way in the NOVA definition. Should they be? And then the other challenge with this heterogeneity of exposure is what that ratio is when people are using these relative gram and calorie measures. So I told you that for gram measures, the ratio is largely influenced by beverages, ultra-processed beverages in the numerator, and unprocessed beverages like coffee and tea. And when studies capture water, that is a huge contributor to the denominator. Whereas energy on the left, the biggest contributors we get to the numerator are things like breads. And on the denominator, unsurprisingly, is things like fruit because they're heavy, right? They have a lot of gram weight when consumed. Okay. More challenges for studying UPF cancer associations. It was already said today that the type of research I do, epidemiologic study, it's observational. We cannot prove causality, but we can be very intentional and rigorous in how we conduct our studies so that we can infer or better infer causality, ask better questions, you know, make better hypotheses. And so what my team really tries to do is to really up the rigor, up what we know about cancer etiology to really support the observational data that we have. And the main question here is, are we observing real biological signals or artifacts of exposure misclassification? So we always consider measurement error, conceptual ambiguity. We really want to make sure that our definition is clear. And then, of course, confounding. And I couldn't miss the opportunity to show you all a DAG. Those pink bubbles up at the top, those are the typical confounders we might put in a model for cancer. But then what I have visualized down here for UPF and cancer, and fill in whatever, you know, cancer type you want, but what are the potential mediators? Is it diet quality that's mediating the association? Is it body mass index? You know, these are things that we usually can observe in our studies. Is it microbiome? Is it contaminants or additives? These are things that we do not observe in our current studies, and so we really can't draw any causal inferences about. So estimating UPF exposure. So my group is doing this in 2 ways, by self-report and with biomarkers. And the main question that we want to answer is, can we measure usual exposure, right? Not acute exposure, but what is your exposure to UPF over a long period of time, because cancer develops over a long period of time. And we want to know if our tools and our measures are valid. And this is essential because before interpreting associations with disease risk, we need to understand how well we measure our exposure. So in the NIH AARP Diet and Health Study, we have over 500,000 adults. They were middle-aged or older at baseline with a mean age of 62, and the study was started in the mid-90s. And the states and metropolitan areas highlighted in blue is where AARP members were recruited from. We collected baseline cohort information, 125 item validated food frequency questionnaire, demographics, health and lifestyle, and then we have more than 20 years for cancer incidence follow-up. And this is key because consistency is key and validation is key, right? Those are core principles. So before running a single risk model, we wanted to carefully operationalize our exposure and validate it. So we were able to do this because in AARP, there's a sub-cohort of 1,900 individuals with serial 24-hour dietary recalls. And the group that developed the NOVA system, they did this with 24-hour recall data. And so what we thought is, well, we don't have to actually classify anything. We will just use their database, which they generously shared with us, and we will just use their classifications. So agree with it or not, we can say it's NOVA defined because we're not classifying anything. We're using their classifications. So this follows the methods, the approach that were outlined for N. Haynes by Eurydice Martinez-Steele, who's a collaborator from Nupins in Brazil. So then in the full cohort, now we don't have the 24-hour recall data. We have an FFQ, but we were able to use this approach of disaggregating the composite FFQ items. So we take a composite food item like breakfast cereal, and we can say, okay, there's 50 underlying items in a nationally representative population used to develop the FFQ that go into breakfast cereal. We weight them appropriately. We link it to the nutrient database, and we can get sort of this like composite of breakfast cereal where we're saying some of it might be NOVA 1, some of it might be NOVA 4. And so that's what we did. So we linked the 204 line items on the FFQ to 3,500 FNDDS food codes, which linked to thousands of standard reference codes. And at that point, we linked to the NOVA database created by our colleagues in Brazil. So we were linking the SR codes to the NOVA classification. We also did an expert consensus method just to sort of see what other cohorts have been doing for comparison, but I'm not going to present that today because we always use our disaggregation method. And then we validated in this sub-validation cohort. So we used two metrics, a validity coefficient, which is essentially just a correlation coefficient between the true intake as estimated by the reference instrument, which is the 2 24-hour dietary recalls using a measurement error model, and the observed intake in the FFQ. And then we also estimated an attenuation factor. This ranges from 0 to 1, and it is the slope of true intake on observed intake. And translating this to plain English, it means if you have an attenuation factor of 0.5 and you have a true relative risk between your dietary exposure and your outcome of 2, if your attenuation factor is 0.5, you're only going to observe a 1.4 relative risk, right? You're attenuating toward the null because of that measurement error, right? So this helps us to be able to interpret our observed associations. Okay, so in prospective studies, measurement error, likely non-differential, it's going to bias associations toward the null. And so this is always coming up in the discussion. We say, well, we didn't see anything, but maybe it's just because it's biased toward the null. Well, let's address it. Let's, tackle measurement error head on. And that's what we try to do. So for our validation, you can see here for the gram and kcal variables for all of the NOVA groups with the NOVA 4 UPF bolded, we have very good results. So I'm just showing the attenuation factors here, but they're all above 0.5, which means realistically for a modest association with a large enough sample size, we should be able, particularly for energy adjusted gram variables, we should be able to observe an association. And so this is what informed our decision in our analyses to focus on energy adjusted gram intake. And then also just to put this in context for the original validation of the AARP FFQ that looked at all the micro and macronutrients and food groups, the attenuation factors ranged from 0.24 to 0.68. So we're measuring UPF, on the upper end of what the capabilities of an FFQ can do, which was actually a surprise for me. I didn't think the FFQ was going to perform this well, but it did. All right, so what does percentage gram intake look like in AARP? So you can see, unsurprisingly, the top contributors to the numerator to UPF are carbonated soft drinks in purple and other sweetened beverages in green. And if we look at calories, again, as I said before, like what they saw in UK Biobank and EPIC, now we have the major contributors being bread and baked goods. Okay, so how are we improving the study of UPF cancer associations? Our goal is to apply the NOVA classification to FFQ data consistently. We have the limitations I mentioned, but our solution for consistency is this linking of SR codes. We did the validation. This is important for the rigor, and it also means that we can run regression calibration when we see an association and try to estimate, what is the real association in the absence of measurement error. And then we're trying to think about this application of energy versus gram and what's contributing to high intake and really trying to dissect that. So let's jump into some data. So the data I'm going to show you today is for a number of obesity-related cancers: endometrial, ovarian, kidney, postmenopausal breast, and colorectal. Endometrial is colored here in pink. It is the strongest association with obesity, with anywhere from like a 2 to a 5-fold increase, depending on the study that you look at, for obesity versus normal BMI. Kidney would be the second strongest, somewhere around a 1.8, 2-fold increase risk for obese versus normal BMI. This varies by histology. And then much more modest associations for the other cancer types. So less strong association. So for endometrial cancer, we started here. This was led by my former research analyst, Caitlin O'Connell. And what you can see is in the main model, we see this association between higher UPF intake in energy-adjusted grams per day. But when we adjust for BMI, it completely attenuates to the null. Now, I said one of the things that we want to do is we really want to introduce rigor. We know quite a bit about endometrial cancer in etiology. And we know that type 1 subtype of endometrial cancer is more obesity driven, and it's different than type 2 endometrial cancer, which is different in terms of etiology from other epithelial cancers. So we look by subtype, and type 1 has an even stronger association with UPF. This is in line with it being a more strongly associated obesity related cancer. But again, once we adjust for BMI, we attenuate our association toward the null. Now for type 2, this is a much rarer cancer, which is why you have such a large error bar here, because it's based on relatively few case numbers. The associations weren't significant, but it was interesting to see a suggestive positive association that was not fully attenuated by adjustment for BMI, suggesting maybe there is something else about UPF if we had bigger numbers that might be contributing to type 2 endometrial cancer. But the main takeaway from this, endometrial cancer is associated with UPF, but it's 100% mediated by BMI. Ovarian cancer, we looked at in the same paper because of that link that had been shown in the UK Biobank. And unsurprisingly, because again, this is not a diet-related cancer, we saw no evidence of an association. And we had almost 130,000 women and many-fold higher cases, 1,200 cases compared to the 100 or so cases that were in that previous study that showed a positive association. Again, we looked by histologic subtype because we know endometrioid ovarian cancer is the obesity-driven type of ovarian cancer, but it's a weaker association. We didn't see anything with UPF. Looking at renal cell carcinoma, which was led by my current fellow, Dr. Kaylin Burns, who's here today. Again, renal cell carcinoma is pretty strongly associated with obesity. And when we look across quartiles, we do see this increased association for renal cell overall. And then clear cell renal cell carcinoma is the obesity-driven subtype of renal cell carcinoma. We see a stronger association. When we adjust for diet quality using the healthy eating index, we don't see much of an attenuation. So this does not seem to be driven by diet quality. But when we adjust for BMI, we see an attenuation. The attenuation makes the overall association not statistically significant, but it does not fully attenuate the clear cell association. So maybe there is something going on between UPF and clear cell renal cell carcinoma. We need more studies. We need mechanistic evidence and biologic plausibility to tease this out. For breast cancer, I'm showing the overall association. Again, it's small. It's that top one up there. No association, whether adjusted for BMI or not. And you can see the subgroups for UPF are sort of all over the place. But overall, we didn't see an association for breast cancer. But remember that obesity breast cancer association is pretty modest. The one subgroup where there was a inverse association was frozen desserts and milk drinks, which is sort of like, well, what's included in that? It's mainly yogurt. And so we did this analysis where we linked to the food patterns ingredient database, FPID. Again, we were able to link the FR codes and estimate intake of calcium, intake of dairy, and other micronutrients and food groups. And what we were able to show here is that UPF calcium was inversely associated with breast cancer risk. Now, there are prior studies suggesting a link between calcium, higher calcium intake, and lower breast cancer risk. So this is in line with calcium being, protective for breast cancer. All right, last but not least, colorectal cancer, which is my area of study. For colorectal cancer, this is what is usually cited as being the strongest evidence for a link with UPF. Again, there's a lot of data here. I'm presenting all of the models together for one reason, and that's no matter what we threw at it, we did not see an association. We didn't see it overall. We didn't see it by anatomic location. We limited this to adenocarcinoma histology because that's important. That's what we think of when we think of diet-related colorectal cancer. It's adenocarcinoma. There was nothing here. We looked at joint effects by diet quality. And what you can see here, the diet quality is indicated by the circle, the triangle, and the squares. And if you look at the squares, those are poor diet quality, and then the different colors are the quintile of UPF. So across the board with that one blip sort of in the middle, data is not perfect, but we do see this association between poor diet quality and higher colorectal cancer risk. But we don't see anything for UPF, and we don't see much of a joint effect. We do see that compared to the lowest quintile of UPF and the good HEI score, we do see a hazard ratio of about 1.23 for those with the highest UPF intake and poor HEI. But again, we see that same association for Q2 of UPF and Q4. So it's hard to say that this is really being UPF driven. This really seems to be diet quality driven. We did this nutrient food group intake by NOVA group again, because colorectal cancer is the one cancer where we have a substantial amount of evidence that diet impacts colorectal cancer risk. Processed meat is an established carcinogen based on the relationship with colorectal cancer. Whole grains are an established preventative factor, as well as fiber, particularly from whole grains. And then calcium and dairy have also been shown consistently to be associated with lower risk of colorectal cancer. So we thought, all right, let's look at these established nutrients and food groups and see how this plays out, because maybe we have this opposing effect. Maybe the red meat from group 1 is canceling out the whole grains from group 4, and that's why we're seeing a null association. So when we looked by food groups and by nutrients, all of the associations were exactly in the direction we would expect based on the evidence on nutrients and food groups. Whole grains, regardless of UPF or non-UPF source, were inversely associated. Dairy, regardless of UPF or non-UPF source, were inversely associated with colorectal cancer. Meat, regardless of UPF, non-UPF source, associated with higher colorectal cancer risk. This is 100% in line with current dietary guidance for colorectal cancer prevention, which is shown here. This is the FCRR evaluation of evidence for colorectal cancer. So does this let UPF off the hook for colorectal cancer? I mean, I don't think so. It just means that we need to clarify our definition. If the hypothesis is that it's additives like emulsifiers based on, experimental feeding trials like this one featured in gastroenterology, then we need to be able to study the emulsifiers, but we can't do that with an FFQ. We can't do that using current NOVA definitions. So conclusions overall, UPF is not uniformly associated with cancer risk. mediation by BMI is more likely when BMI cancer association is stronger. There are differences by cancer subtype and histology, and these reflect the associations that we have established with obesity. For UPF type 1 endometrial cancer, fully mediated by BMI, but for RCC, it appears that maybe for clear cell, it's only partially mediated, and so we need more studies to confirm this and to establish potential pathways. How UPF is defined matters. I didn't present any of the percent kcal results because they are truly null. They are 1 across the board, so there's no associations there. And null associations with UPF with ovarian, postmenopausal breast, and colorectal cancer. Particularly for colorectal cancer, there may be these sort of opposing effects of, different UPF subgroups. All right, I have just a couple more slides on what we're doing in terms of biomarkers. For this, we're combining an observational study design where we've generated a ton of metabolomics data in a longitudinal study, and we're combining it with metabolomics data from Kevin Hall's ad libitum UPF trial that was run a number of years ago and showed that that 80% UPF diets led to weight gain. So this is our study. It's the iData study. It was a 12-month study. Blood and urine were collected at 2 time points, and dietary recalls were collected at 6 time points, 2 months apart over the 12-month period. We used a machine learning algorithm called lasso regression to derive prediction models that could explain the maximum amount of variation in UPF defined as the average of up to 6 24-hour dietary recalls, again, using linkage of SR codes to define UPF. And this is for percentage energy, because that's what the trial looked at, was a diet based on percentage energy. So for our serum score, we selected 28 metabolites. For urine, we selected just over 30 metabolites. The prediction models were not overfit. We had great performance in both testing and training sets. And our polymetabolite score was associated with our exposure, with our UPF phenotype in serum at a correlation of 0.6 with in the training set and 0.47 in the testing set. Now, for many of you, these correlations might not seem so big, but for a nutritional epidemiologist, these are huge. This is, above the concentration biomarker level. This is quite strong, and stronger than we would expect with self-reported data from FFQ for a similar proxy. When we took these scores into the feeding trial, we saw that we could perfectly distinguish between the phases of the feeding trial within individual, supporting the validity that we are actually capturing a metabolite profile that is changing with intake of UPF. So the last little bit, I said, okay, well, the whole purpose of making these scores is to see if they're related with cancer, right? How do we see if these multi-metabolite scores are associated with cancer? So we have a large liver cancer pooling project led by Dr. Catherine McGlynn at the NCI with over 800 cases and controls. This is prospective, so pre-diagnostic blood. We looked at the odds ratios for quartiles of UPF, and we saw nothing. That was for our lasso regression. So I said, all right, well, let's throw another machine learning algorithm at this. Let's use elastic net. This is going to select more metabolites. It's going to have more predictive value. We still did not see anything. Now, remember, unit matters, right? How we're measuring UPF matters. And we haven't seen any associations in the self-reported literature from percentage energy. So we said, all right, let's try this, go back to the drawing board, make new scores based on percentage gram, and lo and behold, we see that 2-fold risk with liver cancer, suggesting that this metabolomic profile of percentage gram UPF is associated with future liver cancer risk. Lessons learned: measurement is central. Consistency is key. Etiology differs by cancer type and subtype, and this shouldn't be ignored. BMI plays a mediating role for some cancer types, but not all. And biomarkers may in the future help to clarify some of the signals. There are lots of opportunities to better harmonize UPF definitions, to use better assessment tools, to integrate with mechanistic studies. We need to be able to distinguish processing from formulation, and hopefully one day create targeted biomarker panels to really have true reproducibility across studies. Some of the questions as I was putting this together that I wanted to sort of present to the group, if we have time for question and answer. I won't read through all of them, but I think one that is particularly interesting to me is like, how do we study additives? You know, if this is chemical carcinogenesis that we're thinking about, how do we measure the additives? How do we measure the contaminants, the contact contaminants like microplastics? How do we account for mixtures of foods? You know, this is very, very complicated science. Where do biomarkers fit into all of this, and will they be able to generalize across populations and platforms? You know, epidemiology suggests that there is maybe something between NOVA-defined UPF and some cancers, but associations are inconsistent. And so we really need the mechanistic studies to test causal pathways, to identify components of UPF that underlie associations, and to establish biological plausibility. And Dr. Erin Hengist is going to talk more about that. So I went a little bit over, but I hope that it'll lead to lots of questions and sort of thoughts about how this is playing out in epidemiology and how we're trying to solve some issues.
Why don't we take and see if there are a few questions now? Thank you. You have given us a lot to digest.
I know. I like data.
No, it's very impressive what you've done. So, yes, please, Erin.
Hello, can you hear me?
Yes.
Great. I think you partially answered it towards the end, but you mentioned the potential discrepancy with grams per day and calories per day, or percentage of calories. I was wondering if you preferred one over the other, or had any thoughts about that. Most of the slides you presented were grams per day. Do you usually analyze both if you have it available?
Yes, so we always analyze both, and we stick the percentage calories in the supplement, which is what most other studies do as well, because there's usually nothing to see there, or sometimes there's kind of funny associations that don't make much sense. So percentage calories we look at because in the descriptive epidemiology, we're almost always talking about percentage calories from UPF, right? That's how they describe intake at the population level. That is a convenient way to describe it. If you talk about percentage grams from UPF, it ends up being a much smaller percentage, something like 20%. And that has to do with, you know, the weight of whole foods versus ultra processed foods and everything else. But there's this discrepancy between how UPF intake is being described in the descriptive literature versus how it's being operationalized in outcome studies. And that's not just cancer studies, that's cardiometabolic outcomes as well. So, you know, most people are prioritizing grams, and actually most are using the main results as percent grams. I didn't mention this. We use absolute grams energy adjusted as our main analysis, but you usually get slightly stronger results if you use percentage grams. And that is probably because the relative measure is also sort of accounting for that dietary pattern of higher UPF, lower unprocessed foods.
I sort of feel that your last set of questions, I appreciate you putting up those questions, but to me, it raised the question in my mind, are we chasing UPF and we should be chasing something different? I don't know if you have some thoughts about that. I think in terms of how do we make the dietary guidelines for Americans better? And what kind of approach to look at UPF or look at the food categories that you highlight? Which one is going to help us make the guidelines better?
I mean, I think, again, this comes down to the need for more mechanistic research, which I know is being done. But at least when we think about cancer etiology, the mechanistic research is really going to fall into two bins. You're going to have your metabolic route of carcinogenesis, so the mechanisms that lead to higher energy consumption and weight gain and obesity, right? That is a potential mechanism by which somebody might develop cancer. But you also have this potential for chemical carcinogenesis, which there's no evidence there, maybe not no, but there's very little evidence there in epidemiology. But I actually think that when people read about UPF and cancer, I think they're thinking very often of that chemical carcinogenesis, and we need that mechanistic data there. I mean, I think we could start by looking at biologic plausibility. Emulsifiers is a good place to start. If they're disrupting the gut microbiome and leading to gut inflammation, you know, how can we study that in population settings? I mean, the truth is we need to collect and biobank stool samples to study the gut microbiome, measure local inflammation, not just systemic inflammation, and do those molecular epidemiologic studies. But we need the investment and the biospecimen collections.
Quick question.
So, Britt, one last question.
Just one quick question. Maybe it's quick. I'm interested in your metabolites and your chemical carcinogenesis and the absolute grams, you know, which would then be absolute exposure, right? And when you had your maps of, or when you had your profiles of metabolites and you knew what the gram exposure and took into account maybe absorption, metabolism and such, did you see maps from maybe additives or various chemicals that would have been in UPF that then traced to your metabolites?
So the metabolomics panels that we've been using, it's the Metabolon Discovery panel, but whether it's Metabolon or another discovery or untargeted panel, they are very broad in terms of the chemical classes and metabolites that they capture, right? So they're going to capture metabolites from food, you know, xenobiotics, but they're going to capture a lot of endogenously produced metabolites as well. So for the multi-metabolite scores, we do select some xenobiotics. Interestingly, they are inversely weighted, meaning that they are negatively correlated or negatively associated with UPF intake. And they are things like vitamins and nutrients that come from fruit and vegetables. Sure. And so those multi-metabolite scores, again, they're based on percentage gram, not absolute gram, or percentage energy, not absolute energy. They are really reflecting not just UPF intake, they're reflecting the metabolic profile of a diet high in UPF and low in, you know, fruits and vegetables.
Mm-hmm. Yeah, that's an area that I think is going to be rich for, you know, all kinds of discovery.
I will say, like in the trial data, we looked at all of the metabolites to see which one changed from phase to phase. And we did pick up things like asulfame potassium and things like that, which, you know, were specific to foods that were fed to people during the trial. The problem with those is that they're so specific to a food that they're not going to capture this very heterogeneous array of what NOVA-defined UPF is.
Great, thank you.
Great. I think we better move on. I think there are many more points for discussion. So our next speaker is Dr. Erin Hengist, who's a postdoctoral visiting fellow in the Diabetes, Obesity, and Endocrine branch at the National Institute of Diabetes and Digestive and Kidney Disease. And he will share mechanistic insights from his work there. So, Erin?
Thank you all for the opportunity to speak here today. I was reading, in advance of this, I was reading the paper summarizing the discussions of the former meeting, and it was kind of nice to see that one of the conclusions was that more controlled feeding studies are needed to quantify the relative influence of dietary factors and identify mechanisms that can explain differences in food intake between minimally processed and ultra-processed food diets. So hopefully some of the evidence I'll present today should contribute to that goal. And I'm here to represent the Metabolic Clinical Research Unit of the National Institutes of Health. The data I will show you today are from studies led by Dr. Kevin Hall and Dr. Valerie Darcy in our lab. And it's the first time I'm meeting many of you in the room, so I'll introduce myself briefly. I'm a visiting fellow from the UK, if you can tell from the accent already, and my background is in exercise physiology. So I studied sport and exercise science as an undergraduate, and then I went on to do a PhD at the University of Bath with my supervisor, Professor Javier Gonzalez, investigating the effects of carbohydrate restriction and free sugar restriction on energy balance and metabolism. In 2022, I moved across the pond to join Dr. Kevin Hall's lab, and I'm leading an inpatient study into the metabolic physiology of ketogenic diets. But since then, I've joined Dr. Stephanie Chung's lab, and I've been contributing to a range of studies across the diabetes, obesity, and endocrinology branch, and have joined, I'm looking forward to sharing some of that data with you. So I don't have any disclosures before I continue. And like I said, I work at the NIH Clinical Center in Bethesda, so it's about 30 minutes away on the Red Line. It's the world's largest research hospital with around 1,600 ongoing clinical trials. And every patient who attends the hospital is admitted to a research protocol. So most protocols I work on recruit healthy volunteers. So when we say patients, it's not just people with cardiometabolic disease. And all of the data I'll be presenting today are from healthy volunteers, as we would call them. And like I said, today I'll be presenting data from studies presented at the Metabolic Clinical Research Unit, or the MCRU, which is a 7-bed unit with 3 additional whole room calorimeters. It has specialist equipment to perform mechanistic physiology studies and is co-directed by Stephanie Chung and Kong Chen. And it's staffed by specialist nurses who I can say firsthand go above and beyond to conduct these studies and really are the unsung heroes of this kind of research. So just wanted to highlight the important work they do. And we work closely with the nutrition department, who are experts in designing metabolic diets for controlled feeding studies. All of that to say, the MCRU is a fantastic resource, and if anyone wants to visit, please get in touch, and hopefully we can arrange that. And it's a resource that is funded by the taxpayer, so we have a duty to kind of do studies that are high priority. This is important context for the evidence I'll be presenting today, because my title mentioned mechanisms. And that can mean physiological mechanisms, but in my opinion, it can also mean dietary mechanisms. So manipulating aspects of the diet that lead to changes in energy intake. We can house participants and manipulate aspects of the diet in the MCRU to obtain kind of unparalleled experimental control of the food environment. And as such, the studies we perform are kind of at the right end of this research continuum. And these types of studies prioritize control and precision over ecological validity and sample size. So hopefully this kind of complements the evidence that Dr. Lotfield has just presented to us as well. So the studies I will present today are kind of, in my opinion, one tool in an arsenal of tools that we can use to understand the physiological mechanisms of ultra-processed food. And I think we need studies from the whole kind of continuum to form a solid evidence base for policy. I promised Marie I wouldn't spend too long on this, but one of the studies I'm presenting today is a direct follow-on from the randomized crossover trial led by Kevin Hall, published in 2019, which compared ad libitum energy intake on diets which are ultra-processed or minimally processed. So participants resided in the Metabolic Clinical Research Unit for 4 weeks and consumed 2 weeks of an ultra-processed food diet, followed by 2 weeks of a minimally processed food diet in a random order. And despite eating to comfortable fullness on both diets, the ultra-processed food diet caused an increased energy intake of around 500 kilocalories per day. And that corresponded with changes in body fat across the 2 weeks. Body weight, sorry, across the 2 weeks, and correspondingly changes in body fat. So why did this happen? Well, this study led to a number of hypotheses around what was driving the effects. For example, eating rate and energy intake rate were higher in the ultra-processed food diet. And also in a post-hoc mediation analysis in collaboration with Dr. Tara Fazzino, members of our team looked at meal level predictors of ad libitum energy intake. And two factors that had the strongest effect size were non-beverage energy density and hyperpalatable foods, as defined by thresholds of paired nutrients, which I think most people here. That was I hope that wasn't a jingle for hyperpalatable foods. But yeah, sorry. As defined by thresholds of paired nutrients, which I think most people here are familiar with, so I won't go into detail just now, but I'm sure we will discuss more later today and tomorrow. So this led Kevin and our team to design a follow-up study, and the primary aim was to determine differences in mean daily ad libitum energy intake between 4 one-week test diets. Participants were randomized to receive the diets in one of 4 sequences. And the data were analyzed using mixed effects models with diet and sequence order as fixed effects and participant as a random effect. One diet was minimally processed and it was low in non-beverage energy density and hyperpalatable foods, according to those thresholds. Another diet was high in, it was ultra processed and it was high in non-beverage energy density and hyperpalatable foods. And these are similar to the original 2 diets in the 2019 study. The next diet was high in energy density, but reduced in proportion, well, in the foods that were determined to be hyperpalatable, and we call that UPF high-low. And the final diet is ultra-processed still, but the non-beverage energy density and hyperpalatable foods were reduced, in line with the minimally processed diet. So all 3 UPF diets had at least 80% of energy coming from UPF according to the NOVA classification. I gambled that Erica would cover that, so thank you. And that's the definition used in this study. But they vary in the percentage of both hyperpalatable foods and non-beverage energy density, as you can see from this table. All diets were served as 3 meals per day, which is different from the previous study because in that study, snacks were also provided, but there weren't any differences in energy intake from snacks in that study. 36 participants completed the study, and we had a wide range of body mass indices and body fat, but no participants had diabetes or other diagnosed cardiometabolic disease, like I mentioned. And participants ate to a similar degree of hunger, fullness, satisfaction, and eating capacity on these 4 diets. And here, just to kind of show the time resolution of these. So this is for hunger. So the resolution of my graphs is a bit low, so I apologize for that. But the visual analog scales were measured just before each meal and periodically in the postprandial phase across waking hours. And here is the same for fullness. So you can see the consistency between the diets. They rated the meals similarly in terms of pleasantness and familiarity. And I put an asterisk there because there was a small difference between the minimally processed and ultra processed high diets, which I think will be interesting to discuss later. And here's the primary outcome of ad libitum energy intake. So in the minimally processed diet, participants ate just under 2,500 kilocalories per day. And on the ultra-processed food, high in energy density and high in hyperpalatable foods, participants ate around 950 kilocalories per day more. When energy density is kept high, but hyperpalatable foods are reduced, there is a statistically significant but subtle reduction in energy intake of around 130 kilocalories per day. And when you decrease energy density in addition to hyperpalatable foods, that results in a further 660 kilocalories per day decrease in intake. And the difference between the minimally processed and the ultra-processed diet that is low in density is not statistically different and is within around 130 kilocalories per day. So this suggests you can essentially normalize the energy intake of an ultra-processed food rich diet by manipulating the energy density and proportion of hyperpalatable foods, according to the definition, with reductions in energy density explaining a large proportion of this effect size. Unlike the first study, we did not observe differences in eating rate between the diets, which is interesting. However, we did observe differences in energy intake rate, which suggests that reducing the energy density and hyperpalatable food drives reductions in intake primarily by this mechanism, and not through eating rate per se in our test diets. And I think we're going to hear more evidence around the potential effects of eating rate as well. But it's interesting it wasn't seen in our trial. Body weight changes corresponded with the changes in energy intake between the diets. So similar to the first study, participants gained weight on the high energy density diets and lost weight on the 2 low energy density diets, irrespective of processing. So this is proof of concept that diets high in ultra processed food don't necessarily cause weight gain. And this is the same data drawn as a change across the 7 days. So you can see here in the red, the minimally processed food, and in the green, the ultra processed food, low, both cause reductions in body mass. However, only the minimally processed diet led to body fat loss measured by DEXA, which suggests there are potential differences in digestion, nutrient partitioning, and body composition caused by highly processed diets that we don't fully understand yet. And potentially some of these effects could be mediated by reducing gut microbial diversity and inducing intestinal inflammation. And 2 of our collaborators at NIH, Dr. Catherine Mackey and Dr. Michael Sack, are actively following this up in samples from this study. I encourage anyone who's interested in this potential mechanism to go and read their commentary in Nature Reviews Immunology to hear more about what they're planning. The participants also resided in a metabolic chamber for 24 hours for 1 day on each diet. And there was an increase in energy expenditure around 120 kilocalories per day between the minimally processed and the ultra processed food high. Again, I apologize for the graphs. And this was primarily driven by sedentary energy expenditure, which we define as sleeping metabolic rate and awake and fed thermogenesis combined. So it's likely explained by subtle differences in body weight and energy intake on that day. So to summarize so far, highly processed diets increase energy intake and body fat compared to minimally processed diets when energy density and proportion of hyperpalatable foods are higher. Reducing the energy density and proportion of hyperpalatable foods of a highly processed diet can essentially normalize energy intake in line with a minimally processed diet. However, there are potential divergent effects on nutrient partitioning between low density, highly processed and minimally processed diets that may impact body composition. And I think this is an interesting area for future research. One aspect of these studies is that they're extremely time-consuming, expensive, and require huge commitment from the participants. So we have an ethical responsibility to build in exploratory aims and research questions within the larger clinical trials to maximize the value of the resources and effort put in. And one of these questions is looking at metabolic responses to meals during these diets. And at the end of each test diet, this is what we did in this study as well. So at the end of each test diet, participants consumed a fixed energy mixed meal tolerance test with meals that were representative of each diet in terms of density and proportion of hyperpalatable foods. So after an overnight fast, participants were provided with the meal test. And like I said, energy content was matched. It was matched at 30% of baseline estimated energy requirements using resting energy expenditure. And that came out to around 780 kilocalories. And blood samples were obtained periodically over 3 hours at baseline and then following ingestion of the first bite. The 4 test meals were reflective of the diets, like I said. So the minimally processed low had an energy density of 0.9 kilocalories per gram and was made up of whole fruits and oats. And the UPF high breakfast used reduced sugar oats and other highly processed ingredients like protein powder and honey roasted peanuts, and had double the energy density. The UPF high-low breakfast was very similar in terms of food and density, but the individual items were not hyperpalatable according to that definition. And UPF low-low similarly had highly processed ingredients, but was low in energy density, similar to the minimally processed food diet. And just to give an idea of what those meals look like, so the minimally processed on the left and the ultra-processed high on the right. And then the high-low and the low-low on the left and right. In terms of the postprandial responses, so glucose and insulin peaked at around 30 minutes following ingestion of the meals before returning near to baseline by 180 minutes. And whilst it looks like there might be a subtle effect for glucose and insulin total area under the curve following those meal tests, there were no significant differences when adjusting for multiple comparisons. Fasting triglycerides were elevated in the high density diets. Oh, I'm sorry. Can you see that? Yeah. Compared to the lower density diets. But the postprandial increase was proportional to the amount of fat in the meal, which you would expect. And as they were matched, that makes sense. So the fasting-free fatty acids were subtly different, but were suppressed in all conditions following the ingestion of the meal. So triglyceride area under the curve was highest after the ultra-processed food high condition, and free fatty acids were lower in the ultra-processed food high condition compared to the minimally processed diet, or meal, sorry. We have also measured gastrointestinal hormone concentrations, which will hopefully complement work being done on the gut microbiome and inflammation, as I previously mentioned, to try and get an understanding behind potential changes in digestion and nutrient partitioning. But yeah, I think, so these data show that diet high in energy density, a high energy density ultra processed food diet increases postprandial triglycerides. And I'm going to segue slightly here because I want to move on to a different study design. But in animal models, this metabolic response has been shown to be a driver of food choice orchestrated by the brain. So what do we know about how these changes in post-ingestive metabolism could impact eating behavior orchestrated by the brain? Well, dopamine is a neurotransmitter that is heavily implicated, but human positron emission tomography or PET studies have so far been equivocal in terms of whether there's a main effect of post-ingestive nutrients on measurable dopamine changes in the human brain, and whether obesity dampens the dopamine responses to nutrients, as some have suggested, albeit with limited data. And I should preface this by saying this is not my area of expertise. I'm representing Dr. Valerie Darcy here, so I'll endeavor to report this as accurately as possible. And this is where Dr. Valerie Darcy comes in. So she sought to test this using an inpatient feeding study, which had more than double the sample size of previous studies. And she wanted to measure people's dopamine responses, well, dopamine before and after a highly processed, high-fat, high-sugar milkshake. Participants were 18 to 45 years old with a wide range of body mass indices and body fat. And based on the limited human data, she hypothesized that D2 binding potential would be reduced following the milkshake ingestion, and that this would be dampened in people with obesity, as others had hypothesized. An important point here is that the raclopride tracer that was used in the study works by displacement. So more dopamine from the milkshake would cause a decrease in PET signal. So D2 binding potential and dopamine release are inverse of each other. And decreased binding potential is measuring dopamine release. So what did she find? Well, contrary to expectations, there was no significant striatal or dopamine response before versus after the milkshake. And this picture is the same no matter where you look across the striatum. but there's some variability between people. And this graph shows the kind of within subject responses pre and post milkshake, as well as the distribution of the overall striatal dopamine binding potential. So was this variability in response to milkshake related to adiposity? And remember here that dopamine release, a decrease below 0 indicates a dopamine release on this graph. That doesn't seem to be the case either. So body mass index was not significantly associated with dopamine response. And this paper has received a lot of attention and debate from the scientific community with follow-up letters and responses. So I encourage you to read that if you're interested in this mechanism. And Valerie and her team are now in the process of testing the responses to the later post-ingested period based on some pilot data they have. This is when differences in postprandial nutrients postprandial metabolites are more pronounced between the diets. So to add to our conclusions around energy intake and body composition, post-ingestive dopamine signals are not associated with body mass index in the early post-meal period, but delayed post-ingestive signals warrant further investigation. And so as we're here to discuss, I wanted to finish with some personal thoughts. I think there's generally consensus around many of the health impacts of highly processed foods, but I think there's a lot of ongoing debate around why and which definitions to implement. And I think personally, I think we're at a fork in the road with regards to policy between restriction or reformulation of ultra-processed foods. But I think it will be almost impossible to remove ultra-processed foods from the food system. So I think that indicates which way we're likely to move. And I think we have good evidence that energy density and energy intake rate are two characteristics that can be targets for reformulation. I think an interesting point is how will guidelines fit in within existing nutrient guidelines. So for example, in the UK, we know there's legislation on foods high in fat, salt, and sugar, but there's quite a lot of overlap between foods that are Nova 4 and HFSS. How would we design studies to kind of isolate the contributions of those? And is it beneficial is it additive at all? And more work needs to be done to understand the overall health impacts rather than just effects on energy balance and obesity risk based on the nutrient partitioning and body composition data that I presented. So on that note, special thank you to all the people who contribute to this work and the MCRU nursing team, to the participants who give their time and effort and bodies and samples for our studies, and thank you all for listening.
I do have some questions. I wanted to be sure I understood. When you give that test meal at the end of the 1 week feeding period, you're giving a test meal of the same that the person was on?
Yes.
At that point?
Yeah, so the meals were designed to be, sorry?
Oh, I was asking about the test meal and its composition relative to the feeding period.
Sorry, I should have mentioned, yeah, the macronutrients were matched between the 4 diets and the energy was matched, but the characteristics of the meal was what was attempted to be manipulated in line with the diet, the overall diet.
Right. And then just related to that, in the first study with Kevin Hall, he did 2 weeks and it looked like some things might have been coming back to a baseline. And so I'm curious why this study used 1 week rather than 2 weeks. If anything, it seems like it should have gone longer.
Yeah, I think that's interesting. I'll preface it by saying I wasn't involved in the design of this, but yeah, I think if you were to do that and keep those 4 groups, you would need to do an 8-week inpatient study unless you did a washout period. So that comes with its own challenges. And then, potentially reducing the groups could make that possible. But yeah, I think it's a valid and fair critique.
Other questions? Erica?
Can you hear me? Really interesting. Thank you, Erin. So I was wondering if in either of the two UPF trials, the 2-phase or the 4-phase, if you have been able to look post hoc at like differences in the weight of the menus, thinking about not just energy density, but when you're eating foods that have more water weight or weight from fiber, if that could also be at play here and explain some of what we're seeing in the epidemiologic data.
So total grams per day. Yeah. Yeah, we could definitely do that analysis.
Question.
Hi, on the UPF studies, so how is the hyperpalatability defined?
So it's defined using Terra Fazino's definition, which has 3 kind of nutrient pairs. I think it's carbohydrates and salt, fat and sugar, and fat and salt. I think there are 3, or I need to check that. So yeah, they're the 3 combinations of foods. They kind of did that based on well, the rationale for coming up with that definition was because it was thought to be very subjective, so they wanted to come up with a way of objectively capturing it using these thresholds. But I think that comes with its own concerns, and I'm sure we'll hear more about that today.
Margaret.
Yeah, so we're going to have a session on this and dig more into the foods in these kinds of studies tomorrow, talking about some of the variability between the diets that isn't what has been controlled for, and the definition of hyperpalatability, which for a lot of us that work in this field is not a very useful term. Because palatability is not a property of food, it's your response to food. So you can't just look at a food and say this is high in sugar, fat, and salt and automatically say it's palatable, because it might not be. But since Erin came into this late, we will leave some of our comments to our session tomorrow. Okay.
Thank you.
Get you off the hook.
Hi, Chris Sully, International Food Information Council. You mentioned something about eating rate and comparing the first study to this study. Could you just briefly recap what the different, you said you might refer to it later. Maybe I missed it.
No, so in the first study, we saw an increase in eating rate, which is grams per minute, and also an increase in energy intake rate, which is kilocalories per minute. So that makes sense. But in this study, we didn't see a difference in eating rate in grams per minute between the diets. So there's an inconsistency between the 2 studies. We don't fully understand that yet. We need to look into that more. But yeah, we can discuss afterwards, perhaps why. But the energy intake rate was different, and that reflects the density of the diets.
What is the status of the second study? Has it been completed and
Yep,
submitted
so
for publication?
it's complete. We're prepping the manuscript, but as you're aware, Kevin's no longer at NIH, so we're kind of yeah, we're working on it.
Just well, first, great presentation. Thank you for sharing. And maybe this is for tomorrow, but you also said something about familiarity that you were going to come back to.
Sure, yeah. If we've got time. So in this study, the minimally processed diet was kind of designed as the control, and then the other diets were compared to that. I think we've seen from this trial now and the free living study by Sam Dicken and colleagues that when you kind of make a diet potentially like healthier, then people lose weight, even if it's ultra processed. So for me, the potential control diet should be ultra processed high, and the familiarity data kind of suggests that actually that was more familiar to the participants coming into the study. So I think that's good evidence for future study designs. and that we should be looking at actually the baseline is high ultra processed food diet, and when we're testing minimally processed foods, that's the bigger intervention.
Great. Other questions? Thank you so much. As we all know, the clinical data are essential for this area to move forward. So it's great to see the progress that is being made. Thank you.